One of the striking features of neuroimaging data and in fact of the human nervous system is the marked inter-individual variability of brain structure, function and connectivity. Really, no matter which feature we examine, we always see a broad range across individuals that is only now fully appreciated, that samples are sufficiently large to actually characterize this inter-individual variability. And so one of the biggest overarching questions in our field is, how do these variances relate to factors that we would suppose are influencing them, such as age, gender, genetics, but also experience, health, and lifestyle? And on the other hand, critically, how do individual variations in brain structure, function, and connectivity then shape individual behavior? So the differences between people in terms of cognitive performance, social affective functions, or psychopathology. When trying to understand inter-individual variability, which is a long-standing topic in cognitive psychology and also neuroimaging, we need to be aware of the limitations of classical within-sample statistics, so t-tests between extreme groups or correlation analysis, because basically all they are asking is the difference between this one group, let's say of controls, and another group of patients that we examined, surprisingly big under the assumption that they actually come from the same population. On the other hand, clinical questions and in fact most real-life applications focus on statement about new cases, so out of sample prediction. Hence, the overarching theme of my presentation, my research, and I think also the field as a whole, is to move away from within sample associative biomarkers towards machine learning based predictors that train a model on existing data and then evaluate it for its capability to make statements about new subjects that the model has not seen before. This talk will roughly be structured in two parts. The first mainly dealing with anatomical structural neuroimaging data, and the second then looking at dynamic networks. So coming to the structural side of things, this is a normal T1 weighted MRI of a young healthy subject. As you see here on the right, there's a second image which is belonging to an elderly subject, roughly 75 years old. And as you can see already from visual inspection, there are quite a few conspicuous differences between these two images. For example, the sulci are very narrow here in the young subject, but substantially wider in the older one. And that also, for example, pertains to the inner CSF rooms. Here, the the frontal horn of the lateral ventricle. Or if you look a little bit more closely, you also see that the gray matter, it is much thicker and better demarked in the young than in the old subject. Can we use this information now for an individual assessment of aging based on brain structure? Here I will demonstrate the general workflow of such procedure. You need to start with a larger group of subjects, several hundreds, if not thousands of subjects, that are scanned with a standard T1 MRI, and the data is then pre-processed in an automated fashion. This is just a small item here on the overall workflow, but I would like to stress that in the context of large-scale machine learning analysis on neuroimaging cohorts, actually the data management, automated processing pipelines, and automated QC are critical aspects. Since I don't have time to go into the details here, for now we will just need to state that we can come from the MRI image as it comes out of the scanner, onto a parametric image that in a standard and hence comparable reference space shows for each subject how much brain, how much volume was at any particular location. We can then condense this information, which is actually natively represented in several 100,000 individual voxels, onto a low-rank representation based on biological information. And the key here is to realize that these 400,000 individual image points are not the actual effective resolution of biology. Rather, the brain is organized into what long has been termed brain areas, and the fundamental assumption here is that within each brain area, structure, function, and connectivity are relatively homogeneous, whereas they change between areas. So, as shown in this example, we can actually represent the images from several hundreds or even thousand subjects very conveniently as a matrix consisting of subjects here in different rows, and the columns then just represent the mean volume of several hundred brain areas. If we have this feature matrix and our associated target variable, the phenotypes, we can then actually train a model to learn the relationships between the low-rank feature space and the behavioral target, which we can then evaluate on new, previously unseen subjects. Here is an example, it's already a couple of years old, where we did this for a few thousand subjects, and we can see quite well that indeed using nothing more than a 10-minute T1 MRI scan, we can actually predict individuals' age quite well. More precisely, we can predict individual age with a mean average error between chronological and estimated age of roughly 4 to 5 years. What's important is that obviously in normal subjects, normal healthy controls, the brain age deviation, so the difference between the estimated and the true age, is centered around 0. Some subjects are estimated older than they are, some have brains that look younger. If we now move into the clinical domain and, for example, look at subjects with mild cognitive impairment, we see that their brains actually look pre-aged. That is, the brains from subjects with a precursor of dementia on average look about 6 to 7 years older than the subjects actually are. And if we move to actual Alzheimer's disease, so a full dementia, then this advancement in brain aging, the brain age deviation, actually increases to about 10 years. Going more into the details, I will actually show you now a recent study that was published this year in Human Brain Mapping, where we not only re-examined this question with a new methodology and a new sample, but we also focused on one of the big comorbidities of dementia, or rather on a medical condition that has a high relationship to cognitive decline, which is sleep-related breathing disorders. Sleep-related breathing disorders, such as sleep apnea, actually are a very important predictor of cognitive decline. So we were wondering, across healthy controls, patients with mild cognitive impairments, and patients with Alzheimer's disease, whether sleep-related breathing disorders had an effect on gray matter structure and apparent brain age. At the first step, we actually used a classical mass univariate analysis to see, are there significant differences in gray matter structure between patients with and without sleep-related breathing disorders. Now, if we look at the univariate effects, we first of all, and importantly I have to stress, can replicate the well-known effects of both age and cognitive status. Both show a prominent atrophy in the temporal lobes, and in particular for cognitive status we also see lots of areas of the so-called default mode, cingulate areas, but also parietal areas that are actually affected by the progression from normal healthy aging to mild cognitive impairment to Alzheimer's disease. Importantly, however, we do not find any univariate effects of sleep disordered breathing. So we moved to the multivariate case, the brain age prediction, and here we implemented a new and I think very interesting scheme. That is that we do not perform cross-validation within our actual sample of interest, but we trained the predictive model on a set of large cohort databases. This is shown here on the top. There are 9 different databases containing subjects between 50 and 85 years of age. And as you can hopefully see in the left figure, the different databases were quite heterogeneous in terms of the number of subjects and the age distribution. So we implemented an ensemble predictor with a stratified subsampling scheme. Now what's the idea here? The idea here is that we try to achieve an unbiased prediction by training weak predictors on unbiased data, which we could obtain through a stratified subsampling of the overall data set. The final prediction was then achieved by bagging the different weak predictors, and as we can see here from the out-of-bag predictions in that training cohort, this gives us a very good and bias-free age prediction model. In more details, this is the structure of our entire database, and then each individual weak learner was a stratified subsampling, stratified by age, gender, and site. So we gave each individual learner a balanced sample, and if we then predict out of bag the remaining subjects, we get to a mean average error of about 4.5 years, and importantly, we do not see any strong bias in the data set. Last but not least, we applied that model to predict brain age in our actual cohort of interest, consisting of healthy controls, MCI, and Alzheimer's patients, both with and without sleep disordered breathing. Here you can see the individual data points, and if we look then at the, again, brain age deviation, so the difference between true and predicted age, we, not surprisingly, but very reassuringly, again found that for healthy controls, basically the brain age deviation is around 0. So healthy controls are on average predicted to be as old as they really are, which they should be. But it's important to note that this is a complete out-of-sample prediction, not just as part of a cross-validation procedure, but actually prediction onto a new site. So the model did not see any data from the sites that actually supplied the subjects for MCI, healthy controls, and AD here. Interestingly, we did not find an effect of sleep disordered breathing. Looking at the MCI subjects, we confirmed previous results that MCI goes along with an accelerated brain aging. Here it's about 4 years, and again, no effect of sleep disordered breathing. Last but not least, Also, the subjects with Alzheimer's disease were estimated to be a lot older than they actually were. On average, there was about 9 years, but somewhat to our surprise, given the relationship between sleep disordered breathing and cognitive decline, there was again not a significant advancement in apparent brain age in these subjects relative to those that did not show sleep disordered breathing. Also, there was no main effect across all 3 groups and no significant interaction. So where does that leave us? I think this study actually has a lot of very interesting aspects and can almost serve as a role model for further investigations, because we have a complementary view of uni and multivariate statistics. We could show that through a ensemble predictor based on stratified subsampling, we can actually get a good and unbiased brain age prediction that also generalizes to new sites. Here, the sites where our patients came from. Most importantly, however, this study combined a replication of previous effects, both uni and multivariately, which is the effects of age and MCI Alzheimer's disease, with an investigation of a new effect, here sleep disordered breathing. And I think that's something we should really strive for, to make more the norm in these kinds of investigations, because what we're seeing way too much is the combination of a new question with a new methodology, which then quickly leads into the problem that we do not know whether, for example, here a null result is attributable to the methods or the actual underlying biological phenomenon. And the combination of a replication with a new question can alleviate these concerns. As introduced at the beginning, I will now come to the dynamic part of the brain, because one of the big advantages of looking at structural imaging is that it's readily available. It's part of the clinical protocol, that it is relatively robust and easy to handle. However, it has one big downside. It does not capture the richness of brain dynamics that you see illustrated here in this short video. And if you look at it in more detail, you will see that there is not only a lot of dynamics, which is actually displayed here in real time, but also that these dynamics are highly structured. So there are networks of areas that together increase and decrease their activity. So to put that together, we have structural imaging that is very robust, that can tell us a lot about effects, for example, of aging and pathology, but cannot give us a window into these structured, time-varying dynamics. Now, how can we, in a group of subjects, look at these networks in relationship to known task-related processing? So we know from about 30,000 fMRI studies by now that there are networks that are congruently active when you look at aspects such as working memory, language, social cognition. But we also now know more and more, and we need to acknowledge more and more, that the same data analyzed in different ways can actually yield quite different results. That's one of the, I think, most underappreciated studies that's already 7 years old by Joshua Karp that looked at the same data just analyzed in different ways. And you can see that the location of their findings varied quite a bit. Now, one of the common criticism for these kind of investigations is, yeah, you can basically go through all different kinds of settings, but would anybody really use them in real life? Or isn't that fear-mongering, and in the end, such kind of variability would never be seen in the actual literature? Well, that question was, I think, quite convincingly answered by the recent NAPS study, where more than 70 teams analyzed the very same data set using their standard pipelines. So there was one data set that was provided to all of these teams, and then they should just analyze them for several hypotheses in the same way that they always analyze the data. And the results were honestly rather shocking, because as you can see here, for most of the hypotheses, there was quite a heterogeneity. There were some findings that were made by about a quarter of the groups, some that were made by maybe half, 2/3 of them, and only very few hypotheses were actually answered in the same way, like completely congruent across the different analysis teams. So where does it leave us? Basically, we need to acknowledge that there is a huge variability in the literature, even investigating the same topic. Here's an example. These were the activation foci for about 200 fMRI studies investigating neural correlates of working memory. Two things hopefully stand out. There's virtually no brain area that has not been implicated as the neural basis of working memory processes. But on the other hand, I guess most of you also see that there are definitely some areas that are more frequently reported than others. Can we actually use that to arrive at something like a consensus map of working memory processes? And we can do that through what's been called coordinate-based meta-analysis. So this is a family of techniques, with activation likelihood estimation being the certainly most developed and most widely used one, that is basically asking the question, where do all of these results that are reported in the literature show a significant convergence? The way to do that is to first accommodate spatial uncertainty that's associated with any reported findings in these kind of activation likelihood estimation maps, and then perform an inference against a null distribution of random spatial association. So this procedure brings us from a very big but also very heterogeneous literature onto a set of brain areas that apparently are something like the robust neural correlates of a particular mental process, in the example I just showed, working memory. And importantly, we can now use the peaks of these areas as our best guess nodes. So for a new subject, where do we think the working memory network is most likely located given the literature? And then we can, in a new subject, extract the network dynamics, the connectome, for this particular functional network. Now you may ask, why do not actually delineate the network in that one particular individual? This is certainly feasible, and projects like the Human Connectome projects are providing data to do that. But if our goal is ultimately to go into practical, for example, clinical applications, task-based neuroimaging, individual delineation of activation peaks is hardly feasible. So here we can conceptualize this approach, these network models, as a workaround. We have a lot of task activation data that gives us robust networks, and then we can use this as priors to look at individual single subject data acquired in the resting state. And in my view, that is providing an important bridge between task data that can define functional areas, but it's not applicable, for example, in clinical practice, and on the other hand, resting state analysis, where you can obviously also, for example, through ICA, define brain networks, but lose the relationship to behavioral meaningful tasks. Now the question is, if we look at these networks and look at these network-specific connectomes, can we train machine learning models in the very same way as I've showed for structural data to then infer individual phenotypes? And here's an example where we looked at the emotion processing network, predicting behavioral traits, more precisely predicting personality. And as you can see here, we can actually make, with some confidence, individual predictions about individual personality traits from resting state connectomes extracted from a priori functional networks. Is there some specificity to that? Because you may argue that it might not even matter where we extract the connectome. It may be a very distributed process. Well, here's an example countering that. You see two networks, cognitive action control network and the social affective default network. And for both networks, we again extracted the connectomes. Key point here. It's the same resting state session. The subject has only been in the scanner once. But likewise, as we had different assays, we can extract different proportions of the resting state data corresponding to different functional networks. And if we now predict something like spatial working memory that we can probe through the Corsi block tapping task, we actually find that the cognitive network is quite good in predicting this particular phenotype, whereas we cannot predict it at all from the socioaffective network. And that's a very important message, because it shows that task-related networks that we defined a priori through neuroimaging meta-analysis, that they do allow different views and differentially useful views on the same resting state data set. Here's another example from the clinical perspective, where we looked at a whole range of different networks, asking a very straightforward question. How good is each of the networks in differentiating Parkinson's from matched controls? And how good is the same network in differentiating schizophrenia patients from healthy controls? And what we see here, without going to the details, is two very important things. First, not every network is equally suited to diagnose both disorders. We must note that the red line is the formal significance level, so really there is some information in every disorder, on both disorders in every network, but there are some networks that are very good in predicting Parkinson's disease, like here on the right side, memory and motor networks. Other networks here on the left, empathy, emotion processing, and cognitive control, that are much better in predicting Parkinson's, schizophrenia. And conversely, also for a given disorder, some networks are very good, other networks are relatively poor. Where does this lead us? There is a growing amount of evidence that we can use these kind of different task-based, meta-analytically defined, almost like essays that look into different proportions of a very rich but poorly structured data set. And I will give you a final example showing how you can really develop a whole pipeline from that, and this example deals with schizophrenia, where we recently proposed a 4-factor model of psychopathology, and then basically looked at a whole range of different brain networks and asked each of these networks, can we predict individual levels of different symptom dimensions in schizophrenia patients using these networks? And as you can see here, this is rightfully criticizable as a big fishing expedition. Lots of networks, different outcomes, so we would not be surprised to see a whole range of significant associations. And that's true, and yet untrue, because this is really only the network discovery stage. So here we tested a lot of networks in one particular set of data. It's actually quite a large data set with more than 100 subjects coming from different sites, and found several associations, several predictive associations. So for example here, the extended social affective default network was quite good in predicting individual cognitive symptoms in that set. Here you see the network and the associated phenotype or phenotypical dimensions. But what is critical now is that we actually then went to a completely new independent data set that was not used at all before, and actually tested whether the nominally significant associations between true and predicted actually also hold in that new data set. And there we could show that, for example, this association between the extended social affective default network and cognitive symptoms, they did generalize. It was actually one of the very few predictive models that generalized to a completely new independent data set. Last but not least, so here's the data. So we had 2 completely new sites, Hartford and Baltimore, and there were 6 formally significant associations through the both tenfold and leave one side out validation in the original multi-site data, but only 2 of the 6 associations actually also then generalized to these 2 new independent sites. And as a last step, we actually also performed enrichment analysis looking at receptor distribution patterns in the respective networks. And then there is sort of this whole story coming together that the social affective default network was not only predictive within our original multi-site sample, it could be validated in a new data set, but it also showed then a relationship to both the dopaminergic and the serotoninergic network, both receptors that are actually also used or targeted in antipsychotic medication. So, where does that leave us in summary? I think we are in a very exciting position at the moment of having large databases of imaging, behavior, and genetics, that we're making a lot of progress in data management and processing, and can start to build platforms for machine learning based analysis of variability and brain behavior relationships in health and disease, where we can then build predictive models that can be useful to actually learn something about new, previously unseen subjects. We need to incorporate prior information on brain organization, be it atlasing to reduce the dimension on structural data, or consolidated functional information in the case of dynamics. And as I've shown, I hope I've shown over the course of the presentation, the critical question is always generalization, not just to new data, but to new sites, to completely independent assessments. And in my view, the road to go here is to have very extensive network discovery models where you really go on a massive fishing expedition in your original data, and then you have subsequent refinements. For example, if you have originally multi-site data, you can start with leave one out analysis, but it also always and critically needs to include a completely independent validation data set that is really only investigated once after you have finished the whole refinement process. And with that, at the end of my presentation, I thank you for the attention. I thank our funders for their generous support. Obviously, everybody in our institute, and I'm very happy to take questions now. Thanks a lot.